Okay, hi everyone, this is Lilit once again, and I am one of your IAs for BIBC 102. In this video, I will go over Lecture 4 and Lecture 5 of Week 2. Before we start, something important that Professor Hampton also talked about in class is the difference between rate of the reaction and its spontaneity. Rate of the reaction simply tells us how fast the reaction takes place. However, spontaneity tells us how far the reaction goes to the right, meaning how much product is actually made through the reaction. So make sure you don't confuse rate and spontaneity of the reaction. Now on to new material. We talked about coupled reactions, and what do coupled reactions mean? Coupled reactions are reactions that help one another. As in, if we have a reaction that is spontaneous, meaning it has a negative delta G, it has the ability to drive another reaction that would not run by itself had it not had the help of this more spontaneous reaction. If you'll take a look over here, if molecule 1 is becoming molecule 2 through some reaction, this will not take place unless the energy from ATP hydrolysis is harvested and used to run this reaction. In Lecture 5, I will talk more about ATP and how it actually contains the energy needed to run these reactions. But for now, just understand that these two reactions are coupled because the energy released from ATP hydrolysis into ADP and phosphate is used to drive the reaction between molecule 1 and molecule 2. When we understand this, we can also understand that there are molecules in our body, for example, intermediates, that are very highly energetic, right? They're energy-rich intermediates, meaning when these molecules, these intermediate molecules, are broken down, just like ATP, they release enough energy that is used to drive an otherwise non-spontaneous reaction. And in most cases, this energy that is let out of these energy-rich intermediates is when a good leaving group breaks away from the molecule. So if you have a molecule and on it you have a good leaving group, when this bond is broken and leaving group leaves, then this energy is used to drive a non-spontaneous reaction. And we will get to this concept more and more as we go through the quarter. Now to move on to redox reactions, it's important to understand what reduced and what oxidized means. To remember, simply remember that when a molecule gains electrons, then it is being reduced, and when electrons are lost from a molecule, then the molecule is being oxidized. If we take a look over here, iron and copper, we can see that iron's oxidation state is 3+, meaning iron is missing 3 electrons. Now let's look over here after the reaction. We see that iron is now missing only 2 electrons because it is only 2 positive. This means that this iron on the reactant side has gained 1 electron, meaning that iron has been reduced. Let's take a look at copper. Copper's oxidation state is +1, but on the product side, it is now +2. Over here, copper was missing only 1 electron, but over here, copper is missing 2 electrons, meaning that from here to here, copper has lost yet another electron. And as we said earlier, electron loss is oxidation. So in this reaction, iron is reduced and copper is oxidized. Now let's break down this reaction into what are called half-cell reactions. Half-cell reactions are always written in a reduced format. And how do we know this? Notice that both iron and copper are given an electron, meaning that they are written in a reduced format. However, both of them can't be reduced, correct? One of them must be oxidized. And how do we know this? We look at their half-cell potential, and the number that is the biggest, meaning most positive, then this is the one, this reaction, that is the reduced one, the reduced half-cell. So simply look at the half-cell potential, and if it is bigger than the other one, then you know that this half-cell is the reduced half-cell. This means that this one must be the oxidized one. After doing that, you can easily construct this. Now, it is important to note that a positive half-cell potential means that your reaction is spontaneous, and a negative half-cell means that it is non-spontaneous. This can get a little confusing because, as you remember before, when we talked about delta G's, we said that a negative delta G is for a spontaneous reaction, and a positive delta G is for a non-spontaneous reaction. But over here, it is the opposite. And how can we prove this? We can prove this by this equation. If we have more of our products, then this fraction will be bigger than 1. And a number bigger than 1 for ln gives us a positive number, meaning that E will be positive if there's a lot of products. And if there are a lot of products, then that makes sense how our reaction is spontaneous, because as I said earlier, spontaneity has to do with the amount of product that is produced. Now, if we have more reactants, then you see how this fraction will be less than 1. And ln of a number that is less than 1, let's say 0.1, will give us a negative number for this part of the equation. This means that this will be negative. And as we said, a negative half-cell potential will give us a non-spontaneous reaction. And that also makes sense because if we have a lot more reactants than products, our reaction is not favorable to go to the right. It is non-spontaneous. And I have summarized all of that over here. If there are more products, then E is positive and the reaction is spontaneous. If there are more reactants, then E is negative and the reaction is non-spontaneous. A reminder for you, don't be confused and always remember that a spontaneous reaction has a negative delta G and a positive half-cell potential. And a non-spontaneous reaction has a positive delta G and a negative half-cell potential. And now you may ask, well, how are delta G and E related to one another? And they are related through this equation. And this also makes sense because if your E is positive, then this equation tells us that your delta G will be negative because of this negative number, because of this negative sign. So this makes sense how a positive E yields a negative delta G, yielding a spontaneous reaction. Now, where does reduction and oxidation take place in biology? Well, if we look at the simple reaction of glucose breakdown, then we know that this is an oxidation. And how do we know that? We know this because through every step of glucose breakdown, so every time glucose is further broken down, electrons from the broken bonds are added to these electron carriers called NAD plus and FAD. After these electrons are added to these molecules, then these molecules are reduced because we have now added electrons to each of them. And then later in the course, we will learn how the reduced NAD, which is NADH plus H plus, and the reduced version of FAD is FADH2. They go on to the Krebs cycle. They go on to the electron transport chain to produce ATP. But that's way further into the quarter. For now, what you need to understand is that every time there is an oxidation in biology, the electrons that are lost from that oxidation get put on these electron carriers. Now let's look into these electron carriers a little closely. Over here we have NAD, and this whole structure is NAD, but all we really need to understand further is this business end. And why is this called a business end? This is called a business end because this is where, specifically over here, this is where 2 electrons and 2 hydrogens are added, reducing NAD. Now, if we look over here, these 2 electrons and these 2 hydrogens end up making a new bond to this carbon. And then this 1 hydrogen that has nowhere to go, it hangs around over here. So that's why you have NADH plus H plus. And so this is the oxidized form, and this is the reduced form. A quick tip. Think of a hydride, which is H minus, as in a bond, right? So let's say you have R, this is some kind of molecule, and you have a bond to an H. This bond is made out of 2 electrons, and so a hydride would be this. Or I believe this. Sorry about that. It's 2 electrons, not 1 hydride. And so when a hydride is added to a molecule, then you see how it brings 2 electrons with it over here and reduces the whole molecule. Now, if we talk about FAD, this is the huge structure of FAD. But again, there is a business end, and that's what we need to focus on. We need to focus especially on this nitrogen and this nitrogen. And again, as we saw with NAD, an electron with its hydrogen is added to FAD's business end one by one. So at first, this hydrogen over here is added, and then the second pair of electrons and proton is added over here. And so now this structure is the fully reduced FADH2. Now, before we move on to lecture 5, I want to make sure you guys really understand why these electron carriers are important. Think of the electron carriers NAD and FAD. Think of them as a car, and think of electrons as passengers. If a molecule, let's say glucose has a lot of electrons in it, right? Because it has many bonds. That means it has a lot of passengers. But when glucose is slowly broken down, the passengers get off of glucose, and they can't just be left in the cell without being in a car, right? Because they they are too reactive, they need to be transported. So that's why we have these transportation for them. So that's why the electrons, which are our passengers, get to sit on the cars and either be held on them over here as NADH and as FADH2, or they are taken to the electron transport chain later to produce ATP, or they are used as reducing agents to reduce other molecules. Further in the quarter, we will go over metabolic pathways that actually use NADH and FADH and use their hydride to be able to reduce other molecules. So that's just something foreshadowing for the rest of the quarter for you to think about. To move on to lecture 5, first I want to talk about the importance and function of ATP. You can think of ATP as the energy currency of the cell, right? So as Professor Hampton said, this is like the $20 bill of energy in the cell. And what does this mean? This is the structure of ATP. And over here you see that we have these phosphodiester bonds. And these bonds are very energetic bonds. Meaning, if we break this, then a lot of energy will be released. But if we want to form this bond, then we will need a lot of energy to form this bond. Now that you understand that, we can say that we either consume food, like glucose, to make ATP, or we consume ATP already existent in the cells to perform other cellular functions. So basically, ATP is where we get our energy, and also ATP is where we store our energy, specifically in these bonds. Now, to put that concept in numbers, in delta Gs, we can say that ATP's energy of hydrolysis, meaning the energy that is released after we hydrolyze one phosphate off, is negative 30.5 kilojoules per mole. And as you remember, a negative delta G is a spontaneous reaction. Because this is so negative, it can drive reactions that have more positive delta Gs that are not as spontaneous as this. However, there are molecules that have a more negative delta G, meaning that they have a bigger energy of hydrolysis than ATP. So those molecules break down and release the energy needed to drive the formation of ATP. And again, these both concepts go back to the fact that we can either consume other things to make ATP, or we consume ATP to perform other functions. And with that, we can start talking about the first catabolic pathway that we discussed in class. As you remember, catabolism is when we break stuff down, and anabolism is when we build stuff. So let me zoom in. Over here, I have the scheme and the pathway of glycolysis. I will not go over this too detailed because I think Professor Hampton did a great job during lecture, but I will go over as much as I can. To give you a little bit of information before I start, I've highlighted the names of the molecules and the intermediates in pink. The reaction is going to go this way, just so you don't get confused. I have highlighted the usage of ATP by orange. I've highlighted the formation of NADH and the usage of NADH in red, and I've also highlighted the formation of ATP in green. And I've also written the names of the enzymes in blue. So now that you know how to orient yourself, let's go through this. So when we eat a meal, when we eat something that has carbohydrates, we get a lot of glucose in our bloodstream. This glucose enters each cell through its receptors and transporters. And the first thing that happens to drive glycolysis is we use an ATP to form this molecule, glucose 6-phosphate. This reaction takes place with the enzyme hexokinase, and it's a little bit of a weird reaction because the whole point of glycolysis is to break down glucose in order to make ATP, right? But over here, right off the bat in the first step, we are using ATP. So you might be confused. Well, what's interesting here is that in the first few steps, we have a preparatory phase, meaning that we have to use ATP to form high energy intermediates later on, over here, so that we can use those high energy intermediates to actually build ATP. Okay, so first step, ATP breaks down to form ADP. We use the energy and the phosphate that came off of ATP, and we put a phosphate on this 6' carbon. And if you need help numbering, 1, 2, 3, 4, 5, 6. So now we get glucose 6-phosphate. The second reaction is with a phosphohexose isomerase. Phospho, because we already have a phosphate. Hexose, because we have a 6-membered ring. And isomerase, because we're not really changing anything between this molecule. All we're doing is making its isomer, meaning all we're doing is we're changing its structure and we're not necessarily taking away or adding any new atoms to it. So after isomerizing, we get the molecule fructose 6-phosphate. Now your carbon number 1 is over here, 2, 3, 4, 5, and 6. Then the enzyme phosphofructokinase 1, or PFK1, is used to use another ATP and phosphorylate the first carbon over here. So now we put another phosphate group on our fructose, and we get fructose 1,6-bisphosphate because we have a phosphate on 1 and 6. Now we are done with the preparatory phase, and we are entering the payoff phase when we actually will be making ATP. The next enzyme is aldolase, and what aldolase does is it cuts this 6-carbon molecule into 2 3-carbon molecules. And I like this pathway very much because it shows you exactly which carbons belong where. So carbon 1, 2, and 3, and its respective structures are colored pink, as you can see, and the rest are black. So here are the 2 structures, the 2 3-carbon structures that we get. We get a dihydroxyacetone phosphate, and we get a glyceraldehyde 3-phosphate. There is an enzyme that is called triose phosphate isomerase, and what this does is it turns dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, and it works in reverse also. But for glycolysis specifically, we want to have glyceraldehyde 3-phosphate to be able to continue the pathway. So some of the dihydroxyacetone phosphate becomes glyceraldehyde 3-phosphate. The next reaction uses the enzyme glyceraldehyde 3-phosphate dehydrogenase. And a small tip over here, every time you see an enzyme with the name dehydrogenase, you're most likely going to be oxidizing or reducing something in that reaction. So don't forget about your energy carriers. So in this reaction, what happens is NAD, being in its oxidized form, is oxidizing this aldehyde over here. And if NAD oxidizes the aldehyde, then the aldehyde becomes oxidized and NAD becomes reduced. We also have a inorganic phosphate that is abundant in the cell that comes into the reaction and is added to this oxidized aldehyde. This structure is called 1,3-bisphosphoglycerate because you have 2 phosphates on carbons 1 and 3. For your information, I believe this is 1, 2, and 3. The next reaction uses the enzyme phosphoglycerate kinase. And as you may know, the enzyme kinase transfers phosphates. So, finally, this is the first reaction in the whole glycolysis pathway where we actually get to make ATP. What happens is this bond over here is very energetic. And when this bond breaks, then we have enough energy to catalyze the formation of ATP by attaching this phosphate onto ADP. Thinking in terms of delta Gs, the hydrolysis of this molecule, specifically over here, has a more negative delta G than the hydrolysis of ATP. And that is why the breakage of this is able to form ATP. After we do this, we have taken off this phosphate, right? So all we are left with is a carboxylate on this end. And the name of this structure is 3-phosphoglycerate. 8th step is catalyzed by the enzyme phosphoglycerate mutase. And mutase sounds just like the word mutation, meaning that all we are doing is we are changing something to go from here to here, and we're not necessarily adding or subtracting anything from the molecule. And that is exactly right. All we do is we take this phosphate and we move it to this position. So instead of 3-phosphoglycerate, now we have 2-phosphoglycerate. The 9th step is using the enzyme enolase, and it dehydrates this molecule. Specifically, it takes off this OH and a hydrogen from here, and that leaves in the form of water. And what we are left with is a double bond over here. This structure is called phosphoenol pyruvate. And for the first time, we finally have the word pyruvate in the intermediate. And this last step is easy because the name is exactly what happens in the reaction, pyruvate kinase. Again, kinase, we take this phosphate and we hydrolyze this, and it releases enough energy to drive the reaction of ATP formation, and we finally form the molecule of pyruvate. Pyruvate will be a very important molecule, and you really want to know the structure of it, as we will talk a lot about pyruvate throughout the rest of the quarter. One reactions that take place after the formation of pyruvate is the formation of lactate or lactic acid, depending on how this is if it is ionized or not. And how does this happen? We use the enzyme called lactate dehydrogenase, and we also use a reduced form of the electron carrier, NADH. And this NADH carries a hydride that is able to, here let me draw a hydride over here, the negative charge. It is able to, oh, I drew it in the wrong place, sorry. We have a hydride with 2 electrons, and it is able to attack this carbonyl over here, opening this double bond and forming this alcohol. So this reaction is exactly what I was talking about up here when I said that the NAD can be used to oxidize other molecules, just like over here, right? NAD was used to oxidize this aldehyde, but also the reduced form, NADH, is able to reduce other molecules, just like over here when it reduced this acetone into an alcohol. And finally, as I mentioned already, this molecule is called lactate. This is your first pathway of the quarter, and it is important that you understand every part of it and why different reactions happen the way they do. Some things that I didn't cover, but were covered very well during lecture, was the importance and the mechanism of GAPDH, glyceraldehyde 3-phosphate. So make sure you definitely look at that mechanism that Professor Hampton talked about in lecture. And I think that's it. I really strongly advise each and every one of you to start studying for this earlier than the weekend of the exam. I know that a lot of us like to procrastinate. I'm definitely one of the procrastinators in the world, but one thing that I did do right when I took this class was I studied every weekend, and I reviewed the material every weekend, making sure that I know the new stuff, making sure I don't forget the earlier stuff, because the midterm and the final will cover a lot of material, and all of this can definitely easily get lost in your brain. As always, if you have any questions, don't hesitate to email me. My name is Lilit, and my email address is on Canvas. I will try to link my office hours and the password to this video at the bottom, and I will see if I can also post my notes over there so that you guys can have access to it and follow it as you'd like. Okay, happy studying, and let me know if you have any questions or need further clarification. Bye.